Hi, I'm Maxime Christophe from the Structure and Instability of Genomes team at the National Museum of Natural History in Paris. In this poster, I will guide you through my work on an interpretable neural network model designed to better understand how nucleosome position across the mouse genome and how this relates to genome regulation. Nucleosome positioning has been studied since the 80s. Early research focused on the affinity between nucleosomes and specific DNA sequences. However, it has been shown that in vivo nucleosomes are primarily positioned by the cell's machinery and transcription factors. These DNA binding proteins act as chromatin barriers, creating regions where nucleosomes are evicted and stacked in phased arrays on either side. Traditional biophysical and statistical models for nucleosome positioning require many assumptions. To address these limitations, we developed a deep neural network that predicts nucleosome positioning directly from DNA sequence. To better understand how the model makes its prediction and overcome the black box issue of neural networks, we used in silico mutagenesis. This technique involves introducing single nucleotide mutations across the input sequence and observing how each mutation affects the predicted nucleosome occupancy. If a mutation alters the prediction significantly, it means that the original base was important for the model's understanding of nucleosome positioning. First of all, the network achieves excellent performance. We trained two independent models using two experimental assays, MNAS sequencing and chemical cleavage, both of which measure nucleosome occupancy genome-wide. As you can see in the correlation plot, the model's prediction align closely with experimental data. We observe Pearson correlation of zero 0.6 and 0.75 on the test set. Interestingly, the two models agree better with each other than the experiments do, with a correlation of 0.54 versus 0.39, suggesting that the network extracts a common, robust signal and filters out experimental noise. Moreover, the in silico mutagenesis experiments show high agreement between the two models. This concordance allows us to define nucleosome positioning regions, or NPRs, as genomic loci where nucleosome placement is consistently determined by sequence. These NPRs will be the focus of the following section of this poster. We extracted 20 base pair sequences from nucleosome positioning regions and ran de novo motif discovery using the MIM suite. In panel A, you can see the four most enriched motifs along with the percentage of NPRs where each appears across roughly 650,000 sites genome-wide. As expected, the CTCF motif clearly stands out. CTCF is a well-known regulator of genome architecture. It acts as an insulator, and we often see phased nucleosome flanking CTCF-bound sites, which matches what we observe here. Poly-A tracts also appear frequently. Because of their stiffness, they are poor substrate for DNA wrapping and tend to exclude nucleosomes. This suggests they might act as chromatin barriers, much like bound transcription factor. What's particularly interesting is that in silico mutagenesis highlights many microsatellite repeats as strong contributors to nucleosome positioning. Like poly-A tracts, their repetitive nature gives rise to specific physical properties that affect nucleosome stability. One example is the CTCC repeats, which might serve both as a chromatin structuring element and as a potential binding site for zinc finger protein. Finally, the fourth motif corresponds to the canonical binding site of the KLF-SP transcription factor family, including KLF4, which is highly expressed in mouse embryonic stem cells. One key advantage is that neural networks don't rely on uniquely mapped reads, making them ideal for studying repetitive elements that are usually ignored due to ambiguous alignments. In this figure, we show the fraction of various repeat classes that carry at least one nucleosome positioning region. Notably, some repeat families are overrepresented. For instance, long-terminal repeat elements in brown are enriched in KLF-SP binding motif and may play a role in chromatin accessibility. Line elements in pink, although mostly inactive in the current mouse genome, still retain regulatory potential by embedding promoter or enhancer-like sequences. Finally, the B2 family is highly abundant in mouse. It is known to punctuate the genome with CTCF binding sites and may contribute to the establishment of chromatin boundaries. To conclude, our deep learning model accurately predicts nucleosome positioning across the genome and reveals the sequence feature that drives it. Among them, CTCF clearly emerged as a master organizer of nucleosome landscapes. Finally, we show that repetitive elements, often overlooked, act as anchoring points, punctuating the genome with nucleosome positioning regions and contributing to chromatin structure. That's all for this walkthrough. Thank you for your attention.